Hello everyone, I welcome you all to Biology Lessons with Kalyani. In today's class, we will be discussing about structure of some of the important disaccharides and polysaccharides as per the CBSE syllabus. So let's look into disaccharides first. Disaccharides, as we all know, they are made up of two monosaccharides unit. And the two monosaccharide units are joined together by glycosidic bond. And because oxygen is involved in the bond, we call it O-glycosidic bond. Right? Now, how is this glycosidic bond formed? This glycosidic bond is formed when a hydroxyl group of one sugar reacts with the anomeric carbon of the other. Okay? It will be more clear. Let's take the example of maltose here. Now maltose, it is made up of two glucose units. Okay. So this is the first glucose. This is its C1 or the first carbon. It is the anomeric carbon because this carbon has the aldehyde group or the functional group and is involved in the ring structure, right? So the hydroxyl group of the anomeric carbon or C1, it reacts with the hydroxyl group of another carbon in the other sugar unit, right? So in this case, we have C4, which is reacting with the C1 anomeric carbon, right? So what will happen? A water molecule will be lost and condensation of two monosaccharides will take place. As a result of this, a glycosidic bond will be formed between the C1 anomeric carbon of one sugar and C4 of the second sugar. Is that clear? So what type of glycosidic linkage? We can call it alpha 1,4 glycosidic linkage. Why? Because here see, glucose is involved which has OH in the bottom. So it is called as alpha glucose. If OH is on the top, we call it beta glucose. So the bond is called as alpha 1,4 glycosidic linkage. Is that clear? Next thing which we need to remember is that maltose is an example of reducing sugar. Now what is a reducing sugar? We know that if sugars have a free functional group, mostly aldehyde or ketone, then they can convert the Cu2 plus to Cu plus and they are called as reducing sugar, right? We had Benedict's test for this. We have discussed it before. So here see the first anomeric carbon which has the functional group, it is involved in glycosidic linkage in the left hand side. Now look at the right hand side. We have the C1 which is not involved in any glycosidic bond formation. So here we have a free functional group. So this can reduce the Cu2 plus to Cu plus ion. Hence maltose is an example of reducing sugar, right? So maltose, we need to remember three things for this. Maltose is made up of two glucose units. The linkage is alpha 1,4 glycosidic linkage and it is an example of reducing sugar. Is that clear? Let's move on to the second example, that is sucrose. Now sucrose is made up of glucose and fructose, right? See here how the glycosidic bond is formed. Now C1 of glucose makes a bond with OH group present in the C2 of fructose. So it is alpha 1 beta 2 glycosidic linkage. Why alpha? Because glucose is present in the alpha form and fructose OH is present on the top. So it is beta form. So the glycosidic linkage would be alpha 1 beta 2 glycosidic linkage. Is that clear? Now, important thing is that sucrose is an example of non-reducing sugar. How? Look, in case of glucose, the aldehyde group is present in C1. Now this C1 makes a glycosidic linkage and hence it is not free. Now if we look at fructose, fructose is an example of ketose sugar. Now being a ketone, it will have the ketone group at C2, right? Aldehydes will be present in C1 and ketone group will be present in C2. We know that, right? So the functional group that is present in C2, that is also involved in glycosidic linkage. So both the functional groups of glucose and fructose are involved in glycosidic bond formation and hence they are not free. So sucrose is an example of non-reducing sugar, or it cannot convert the Cu2 plus ions into Cu plus. Is that clear? It's very important and it's often asked in the question, right? Next, look into the example of lactose. Now here, lactose is made up of galactose and glucose. Now, here the linkage is beta 1,4 glycosidic linkage. Why? Because OH is present on the top for galactose and for glucose also it is present on the top. See, the C1 anomer, glucose is also present on the top. So, the glycosidic linkage in case of lactose, it is beta 1,4 glycosidic linkage. Is that clear? Now, let's see if lactose is an example of reducing sugar or non-reducing sugar, right? So, look at left hand side. When we see galactose, now the C1 or the anomeric carbon, the functional group is not free. Why? Because it is involved in glycosidic bond formation. Now here, in case of glucose, let's see what's happening. If you look at C1, the functional group, the anomeric carbon, which is present in the C1, it is free. It is not involved in any bond formation. Hence, it can reduce the Cu2 plus to Cu plus. So, lactose or the milk sugar, it is an example of reducing sugar. Is that clear? So, let's now see the table, what you need to remember about this. Now that you understood the structure, so just remember maltose and lactose, they are examples of reducing sugar, and sucrose is non-reducing sugar. What are the type of glycosidic linkage found in all, and what are the monomeric units of the disaccharides, right? I hope disaccharides is clear now. Let us come to the structure of polysaccharides. Okay, we discussed disaccharides, which are made up of 2 monosaccharide unit. Next is oligosaccharide, which are made up of 2 to 9 sugar units. And third is polysaccharides. If more than 9 sugar units are present and are joined by glycosidic bond, we call it polysaccharide. Now, if we consider both animal and plants, the main way in which energy or glucose is stored in our body, or in case of plants, is polysaccharides. Why? Because the polysaccharides, because of their structure, they are osmotically inactive and chemically non-reactive. Because of these two reasons, always the glucose is stored in case of plants as well as animals in the form of polysaccharides. So if we take plants, the main storage polysaccharide is starch, right? So glucose is stored in the form of starch, which is the main storage polysaccharide in case of plants. Okay, now starch is an example of homopolysaccharide. What is homopolysaccharide? If the monomer unit of a polysaccharide is same, or similar units are getting repeated or connected by glycosidic bond, we call such polysaccharide as homopolysaccharide. In case of starch, glucose is the monomeric unit, several units of glucose are connected together to form starch. So it is a homopolysaccharide. Also, because glucose is the monomeric unit of starch, we call it glucan. Okay. Next, let's look into the structure of starch. Now, starch is made up of glucose units, but it can exist in two forms, amylose and amylopectin. In case of plants, about 20 to 30% of starch is made up of amylose, while 70 to 80% of it is made up of amylopectin. Okay, now amylose, it is made up of 200 to 1000 glucose units. What does this mean? It means 200 to 1000 glucose units are connected together by glycosidic linkage in case of amylose. Whereas if we see amylopectin, about 2000 to 2 lakh glucose units are connected together. Right? Now, amylose is a straight chain, or there is no branching. Okay, and the structure is made up of alpha 1,4 glycosidic linkage. What does this mean? Like the glucose units, they are connected to each other by alpha 1,4 glycosidic linkage. We saw the structure of maltose, right? We have seen their alpha 1,4 linkage. So now 200 to 1000 units, if they are connected by alpha 1,4 linkage, they form amylose. Okay. Now this amylose, it occurs as helix. It is a straight chain. No branching is present. So it is formed as a helical structure. Okay. Now, because the structure of amylose is helical, it can trap the iodine molecules inside it. So what happens, if you add few drops of iodine into amylose, it will turn it into blue-black color. Okay. Now let's see amylopectin. Now in case of amylopectin, like amylose, it has a main chain which is made up of alpha 1,4 units, but additionally it also has branches. And how does it, what is the glycosidic bond which is present in the branch? It is made up of alpha 1,6 glycosidic linkage, right? So this amylopectin, when you add iodine to it, it will give reddish violet color. Okay, let's see the linkages in a more detail in the next slide. Let us now look into the structure of starch in a little more detail. Now, this is the structure of amylose. Okay, now look, there are different multiple glucose units. This is one glucose unit, another glucose unit, and likewise. Okay, now how does the glycosidic bond take place? Now C1 of the first sugar, it will react with the C4 of the second sugar. Again C1 will react with C4, and this pattern continues. Okay, now this type of bond makes a straight chain structure which is unbranched in nature. Okay, having alpha 1,4 glycosidic linkage. Is that clear? Because C1 is reacting with C4. OH present in the C1 is reacting with the OH present in the C4. That's why this type of bond is called as alpha 1,4 glycosidic linkage. Okay, this is your structure of amylose. Now let's see what happens in amylopectin. This is the structure of amylopectin. Now main chain, it is same as that of amylose, which is having alpha 1,4 glycosidic linkage. What is extra? Now additionally what happens, this is your carbon 1, carbon 2, carbon 3, 4, 5 and 6. This C6, it makes the OH present here, it makes a glycosidic linkage with OH present in the C1 anomeric carbon of another glucose residue. So there will be a branching which will be seen in the sixth position, and the linkage, because C1 and C6 are involved in the glycosidic bond formation and OH is in the bottom, so the linkage will be alpha 1,6 linkage. Okay, now what is the benefit of this branching? Now see, if branching is there, then at multiple points hydrolysis of the sugar residues can take place. So amylopectin is relatively easier in hydrolysis, and at the several branching points, at the end of the branching points, the sugar residues can be released to provide energy, right? So the structure of amylose and amylopectin here is basically the same, only amylopectin will have a branching at the sixth position and the bond, extra bond, will be alpha 1,6 linkage. So this amylose, it makes up of 20 to 30% and amylopectin makes up 70 to 80% of starch. I hope the structure of starch is clear. Some more important facts about starch. Now this starch, it is synthesized in the chloroplast and it is stored in leucoplast. We have already discussed it is used for storage because it is osmotically inactive and chemically inert. Overall, amylose, the color of your amylose, it will be dominating over the color of amylopectin, and starch overall it will give a bluish black color with iodine. Another important fact is that during mobilization of the starch for energy production, glucose residues are always removed from the non-reducing ends of the outer branches. What does this mean? Whenever the body needs energy from the starch, so what happens from the end portion, okay, which is the non-reducing end, that is your left hand side of your like starch, from there the sugar residues can be released and energy can be mobilized. Okay, the extra residues, it will be added in the reducing end side, and to the non-reducing end side the sugar residues will be released to produce energy. If you want to see more detail about the reducing end and the non-reducing end, I have a video for it. Kindly go and check it. I'll provide the link here. Now let's see glycogen. Glycogen, it is an important carbohydrate reserve in case of animals. That's why glycogen is often referred to be as animal starch. It is present in high concentration in liver, followed by muscle and brain. It is also found in some yeast and fungi which cannot produce starch. Okay. Now coming to the structure of glycogen, it is similar to that of amylopectin. What does this mean? It will have a main linkage, main body, which will be made up of alpha 1,4 linkage, and branching will be present, in which the glycosidic bond will be alpha 1,6. Okay. So what's the difference between glycogen and amylopectin here? The difference is that the number of glucose units which form glycogen is lesser than that of amylopectin. Okay. In amylopectin, up to 2 lakh residues can be present, but in glycogen the maximum limit is 25,000 or sometimes 30,000. So 30 up to 25 or 30,000 residues of glucose will be connected together in case of glycogen. Okay, what is the second difference? The second difference is that branching in case of starch used to take place after 25 to 30 residues, while in case of glycogen branching takes place after 8 to 12 residues. What does this mean? It means that the structure of glycogen has a higher degree of branching than that of amylopectin. Okay, so these are the two differences. Okay, look at the bond here. C1 is making bond with C4, and at the branching point, at the branch point, we have C6, which is making bond with C1. Okay, so alpha 1,4 linkage and alpha 1,6 linkage. Again, glycogen is a type of glucan. Why? Because it is made up of glucose, which is the monomeric unit. I hope structure of glycogen is clear now. Let's look into structure of cellulose. Now, cellulose is the most abundant polysaccharide. It is made up of repeating units of beta D glucose. Until now we read all alpha forms. Now cellulose is made up of beta D glucose. What is beta? I already told. OH, if it is present in the bottom, it is your alpha form. If OH is present in the top, it is your beta form. So here glucose will have OH in the top in the C1 anomer. That's why it is the beta form. Look, this is making a bond with C1 is making a bond with C4 of another residues. So the type of the bond is beta 1,4 glycosidic linkage. Now the structure of cellulose is not branched, and about 6,000 residues of glucose is present. Okay, cellulose is not digested by your mammals including humans, and also this is very important because it is found in cotton fibers. Okay, that's all about cellulose. Let's look into another polysaccharide, which is chitin. Now, in case of chitin, the monomeric unit is very important. Question is often asked from here. It is NAG. What is NAG? NAG is N-acetyl glucosamine. So, this is the structure of N-acetyl glucosamine. Okay. So, what is the type of linkage? Beta 1,4 linkage. So, what does this mean? Again, C1 position, OH is on the top. So, it is the beta form. The C1 is making a glycosidic linkage with C4 of another NAG unit. So the type of glycosidic bond is beta 1,4 glycosidic linkage. Is that clear? Monomeric unit is very important. Should definitely remember it. Structure, if you can remember it. Otherwise, if you remember the linkage type, it's also enough. Now, chitin, it is a homopolymer and unbranched. It is found in the fungal cell wall and also the exoskeleton of arthropods. I hope in this class you are clear with the structure of disaccharides and polysaccharides, how the different glycosidic bonds are formed. You can also check out some additional resources on carbohydrate as well as reducing and non-reducing sugars, which I have uploaded. I thank you everyone for watching this video, and keep supporting me by subscribing to my channel, liking the video, and also please feel free to comment if you have any doubts regarding the topic. Thank you everyone. Have a very good day.